Hello, children. Once again, welcome to my session. Okay. Yes. How are you doing, children? How are you doing? How are you doing? Say hi in chat box. How are you doing? Yes. Great. Great. Good to hear your responses, children. Yes, good. Okay, children, I hope you remember in last class, in yesterday's session, we were discussing biotechnology principle and process. It was session 1, right? So where we were discussing about the introduction to biotechnology, principles of biotechnology, and then we started discussing tools of biotechnology, right? Yes. So under tools, we started with restriction enzymes, isn't it? Yes, I hope you remember, guys. Yes. So today, in continuation of that, we are going to, as a second session, we are going to discuss about restriction enzymes in more. Let us study how to name the restriction enzyme. What is the principle? What are the rules to nomenclature the restriction enzymes, and a little more and more about function of restriction enzyme. Okay. Yes. So first we'll discuss naming of restriction enzyme. That means how you are going to nomenclature, give name to the restriction enzyme. Okay, let us take an example of very well known restriction enzyme, that is EcoR1. This restriction enzyme is repeatedly mentioned in your NCERT textbook, now children. Yes, so let us look at the word EcoR1 here. The first letter, okay, the first letter E always refers to genus from which it has been isolated. In this case, EcoR1, the restriction enzyme EcoR1 is isolated from a bacteria, that is Escherichia. The genus is Escherichia, okay? And the followed by E, you have two letters, co. Always co refers to, I mean to say, after the genus letter, the following two letters are from species to which it belongs to. So in this case, this restriction enzyme EcoR1 belongs to species coli. Hence, from the species name, we have taken first two letters CO, that is your CO in the enzyme name. Okay. And now you can see here third letter R. R represents after genus letter, the letter from the genus name, and then followed by two letters from species, and then this is the letter from strain. So EcoR1 is isolated from Escherichia coli bacteria with strain RY13. Okay, so it is R is the first letter of the name of the strain, EcoR. Okay, yes, and 1. What it is 1? 1 means, say for example, in Escherichia coli RY13 bacteria, there are many restriction enzymes, and EcoR1, this is the first isolated enzyme. So this was the first isolated enzyme, hence it is given the number 1. So this is how we name the restriction enzyme. So 1 refers to the number of the enzyme which is isolated from that particular organism. Okay, children, got it? Yes, any doubts here? Any doubts? I'm looking at the QA panel. Any doubts? If you have any doubts, just let me know there. Okay. Yes, good afternoon. Good afternoon. So any one of you have any questions, just post here, children. Just post here. I'll be looking into the QA panel and help you out. Okay. Yes. So I repeat, while naming restriction enzyme, any of the restriction enzyme, first letter will always from the genus from which it has been isolated, and second two letters will be always from the species from which it has been isolated, and third refers to strain, the first letter of the name of the strain, and the numbers indicates like it is the for example, EcoR1. 1 means this is the first enzyme which has been isolated from Escherichia coli RY13. If it is EcoR3, then it is the third enzyme. It has been isolated from this Escherichia coli strain RY13. Okay, hope you people understood how to name the eco R1 as an example of restriction enzyme. Okay, hope you don't have any doubts in naming of restriction enzyme. So the same way like eco R1, we have BAMH and all, SAM and all, right? So we have so many restriction enzymes. Okay, yes, one here, one, why we have written one, beta? Yes, good question. Okay, so some of you are asking why you have written one. Yes, so eco R1, this is one, Roman numerals one. Okay, so this means this enzyme is the first enzyme to isolate from this bacteria, first enzyme. Okay, this eco R1 is the first restriction enzyme to isolate from this bacteria. Okay, this Escherichia coli RY13 strain. Okay, hence we write the Roman numeral 1. If this enzyme is second enzyme, then we write the Roman numeral echo R2, echo R3. Like that, we indicate the Roman numerals in front of the naming. Okay, I hope this clear your doubt. I hope this clear your doubt, W. Okay, yes. So now let us move to next slide. Let us see what is there in next slide for you. Okay. Yes, children. In case of eco R1, okay, you know that palindromic sequence, right? The restriction enzymes cut the DNA sequences at specific nucleotide sequences, specific points, and those are called palindrome sequence. Those are called palindrome sequence. Here you can see, here I have taken an example of eco R1. Okay, you know the palindrome sequence. The recognition site for eco R1 is GAATTC and GAATTC. You can see here. So 5 prime to 3 prime and 5 prime to 3 prime. So both are palindrome, same sequence in opposite direction, right? Yes. So whenever you use a restriction enzyme like eco R1 to digest a double-stranded DNA, okay, you know it cuts at specific recognition site. After cutting, so after the cut has been made, after the digestion, you get two ends, right? See here in this case, it is cutting here between G and A, between G and A. Once it has been once it has been cut, okay, you get two separate ends. See, earlier it was like this. Now your restriction enzyme has been cut here. So it become two pieces, right? So you are getting two ends. And how the end will be? Can you see here how the end will be? In this, you are seeing overhanging ends. Can you see here? Overhanging, hanging ends, hanging ends. Okay, after digestion with the restriction enzyme, if you get the DNA fragments with hanging ends, then this type of ends are called sticky ends. What are sticky ends, children? What are sticky ends? After cutting the piece of double-stranded DNA using any of the restriction enzyme, okay, you will get two fragments of DNA pieces, right? Yes. So if the end of those fragments are hanging, can you see here? It's hanging here, right? Yes. So if you get like this hanging kind of ends, then the ends are called sticky ends like this. Okay. Can you see here? Sticky ends. These two are sticky ends, overhanging, hanging here. Yes. Sometimes if you use some restriction enzyme, say for example, in this case, I have taken, okay, SMA1. This is another restriction enzyme. Okay. The recognition site for SMA1, small 1, is CCCGGG. Okay. So 5 prime to 3 prime, CCCGGG. Okay. This SMA1 small 1 cuts DNA fragment between C and G. Now see, you're not getting any hanging ends here. Can you see any hanging? No, you're not getting any hanging. It's just blunt. It's just blunt. These are nothing but blunt ends. So what I'm telling you here, whenever you use restriction enzymes to cut a piece of DNA, you will be getting two types of ends. You will be getting two types of ends in the DNA fragments. Okay, two types of ends in the DNA fragments. Either the end may be sticky ends when we use EcoR1 kind of restriction enzyme, or we may get blunt ends when we use SMAR1 kind of restriction enzyme. Okay, so any doubts in sticky ends and blunt ends, children? I'm looking into your QA panel. So tell me, any doubts in the sticky end and blunt end? ओके चिल्ड्रन, आपको कुछ प्रॉब्लम है, आपको कुछ क्वेश्चंस है इस स्लाइड से? आप समझ रहे हो व्हाट इज स्टिकी एंड व्हाट इज ब्लंट इन? Okay, I hope it's clear now. Okay, yes. So now with this information about the ends of the DNA fragments after restriction digestion and types of ends, let us see what is there for you. And next slide. Okay, children, this is very beautiful slide. Just I wanted to show you this picture is from your NCRT textbook. Okay, I just want to show you something here. Children, see, this is a vector DNA. Yesterday we discussed, no? In last session we discussed what is vector. Vector is nothing but some vehicle, right? Vector is nothing but vehicle. So this vector or vehicle transfers your DNA into your host cell, right? It transfers your DNA into host cell. So now to take that, see, vector is a vehicle like your school bus. What your school bus does, it takes you into its seat and it drops you to school. The same way, now imagine this is a vector DNA. See, for example, I'm drawing here vector example plasmid I had taken in last session. No, this is plasmid, a double-stranded circular DNA. Now, just assume this is a one end of the plasmid, means vector DNA. You can see here, this is vector DNA. I just cut it open with the help of restriction enzyme. Okay, this is cut it now. This is cut in. Now you can see I'm using restriction enzyme. I'm enlarging that. Just assume it has been enlarged. Okay, what is happening here? Let us see. So this is vector DNA. In the vector DNA, you have recognition site for EcoR1, that is GAATC, right? GAATTC. Yes. And this is foreign DNA. Let us assume it is a gene for insulin. Okay, now this gene also having recognition site for EcoR1. Now I use EcoR1 to cleave both vector DNA. See, my earlier vector DNA was like this, no? Yeah, so now I want, see, vector DNA is like this. Now I want to cut open to fix it into to fix my foreign DNA into it, right? So now I need to cut. How I'm going to cut? I'm going to cut this vector DNA using restriction enzyme. Imagine I'm cutting it. So now this is the restriction enzyme I used. The same restriction enzyme I'm using to cut foreign DNA also here. Okay, I'm using EcoR1 to cut my plasmid as well as foreign DNA. You can see here, this is vector DNA, this is foreign, my insulin gene. Okay, after cutting with EcoR1, can you see it resulted in sticky end, right? Now assume sticky ends. These sticky ends of foreign DNA and the vector DNA now can be joined, now can be attached, right? Yes, so here I'm doing that job. I'm overlapping this to see this is DNA of vector and this is DNA of foreign DNA. Now I'm stitching it, I'm gluing it. How I'm gluing? See, I'm just stitching here. Okay, how I'm gluing or stitching here? I use an enzyme called ligase. Ligase is an enzyme. It is called molecular glue. Molecular glue. What is the function of this? So, glue fixes two fragments, right? The same way, this ligase acts as your molecular glue. It stitches two fragments. It stitches two fragments by making phosphodiester bond, children. How it stitches the two fragments? It stitches the two fragments by making phosphodiester bonds. Yes, very good. So I'm getting good response from you guys. Good. So now you understood. See, I use the same restriction enzyme. Yes, vector to carrier hai beta. That was I was telling. Vector means vehicle, carrier. Yes, see, here one thing you need to remember. I am using echo R1 to cut both vector and foreign DNA. Why? I'm sorry. See, I'm using same enzyme here because I need same sticky ends such that they will be complementary with each other and they can easily, get attached together. That's why I'm using same end. See, if, in case, imagine if I use SAM H1, small H1, I'm sorry, I'm just getting cough. Okay, see, if I using one type of restriction enzyme like SMAH1 and to cut the vector DNA and echo R1 to cut the foreign DNA, what will happen? One end will be blunt like this and another end will be overhanging. See, blunt end and overhanging end. You cannot stitch, right? So you need two overhanging ends such that when you make them sit one above the other, due to their complementarity, they will join easily. Are you getting the idea behind use of same restriction enzyme here, children? Children? Yes. Thumbs up. Thank you so much for your response. Now see, I have combined. Combined means mixing, right? Yes, here in this picture, what I have combined the insulin gene. Okay. And I have combined insulin gene with plasmid DNA. Yes. So now can I call it as recombinant DNA? Absolutely. This is nothing but recombinant DNA, right? So when you have a foreign DNA in some host cell, then that DNA is called recombinant DNA. So when you have a foreign DNA in some vector molecule, that vector molecule plus foreign DNA gives you recombinant DNA. Is it clear, children? Is it clear? Any doubts here? कुछ दिक्कत है? आपको कुछ समझ में नहीं आ रहा इधर? Good afternoon, children who are joining now. Welcome to session again. Yes, आपको कुछ problem है? कुछ doubts है इधर? Can I proceed? Can I proceed, Akanksha, W, others? Can I proceed? Okay, चलो हम देखते हैं आगे क्या है. So up to here we understood the importance of restriction enzyme. Why we need to use same restriction enzyme to cut your vehicle, that is vector and the foreign molecule. Okay, and we also understood what is recombinant DNA, and we also studied a little about ligase enzyme, which is called molecular glue. See, while your restriction enzymes are called molecular scissors, while these restriction enzymes are called molecular scissors, ligase is called molecular glue because it fixes two fragments. Okay, yes. So with this, चलो आगे देखते क्या है आगे का स्लाइड में आपको क्या मिल रहा है. Like what we are going to discuss in next slide. Yes. So children, now we have a fair enough, fair idea about restriction enzymes as a recombinant tool, RDNA technology tool. Since previous session, we are discussing, we are we are focusing on restriction enzyme as a tool of recombinant DNA technology, right? Yes. So now we got enough information about restriction enzyme as a tool of our DNA technology. Now we are going to study another tool, important tool of our DNA technology. That is nothing but vector. Okay, already we know that vectors are nothing but vehicles, right? Vectors are nothing but vehicles, and vehicles made of what? They are made of DNA molecule, right? So your vector molecules are made of DNA. And what is their function? Their function is to carry. The main function of these vectors is to carry your foreign genetic material. In our case, what is the example for foreign genetic material? We are discussing about insulin, right? Since last session, we have taken example of insulin, like foreign gene, like insulin. Okay, so to whom it is given? To another cell, that is E. coli. In our example, it is E. coli. So vectors are the vehicles which are generally made of DNA, and the main function of these vectors is to carry the foreign genetic material, the gene of our interest like insulin, into the host cell, into the host cell. So here in this example, what is a host cell? It is E. coli, right? Yes. So you know a lot about vector. You now by this time you have fair idea, right? Yes. Then what all can we use as vectors? Vectors may be plasmids or maybe phages. Children, phages means what? Phages are nothing but a group of viruses, right? Group of viruses. Okay. Yes. So plasmids. Children, tell me what is plasmid? Plasmids are extra chromosomal DNA, right? Extra chromosomal DNA you get mostly in bacteria. Okay, so before discussing plasmid as a vector, let us learn some important, characteristics of plasmids. Let us study some plasmid characters, and then we'll come back to the slide. First, we'll understand what are plasmid. I use the word plasmid here, right? So I don't know how many of you are aware of the word plasmid. So thought of giving just 1 minute introduction to plasmid. Okay. Yes. So what are plasmids? Plasmids are, if you look at the bacterial cell here, this is bacterial cell. If you're looking inside the bacterial cell, you can see small double-stranded DNA molecules which are circular. Can you see here? Small double-stranded circular DNA molecules. Okay, these are nothing but plasmid. And children, you know here, this is bacterial DNA. This is main DNA. Okay, this is main DNA. In addition to the main DNA, we have this. Hence, plasmids also called extra chromosomal DNA. Plasmids are extra chromosomal. Why it is extra chromosomal? It is other than the main DNA. That's why it is extra chromosomal DNA. Okay, so plasmids are extra chromosomal DNA. They are in extra. Okay, they are not main DNA. And what is the nature? They are circular and they are double stranded. Okay, yes. And one more important character, plasmids are self-replicating. Self-replicating means they can undergo replication. See, this is one plasmid, and this plasmid can undergo multiplication to form new cells, new plasmids. See, this is parental plasmid, and this plasmid divided into or replicated into new plasmids. So they can self-replicate. They are not under the control of main DNA, main chromosomal DNA. Okay, so they can. Okay, so they can replicate on their own without help of main DNA, without help of main DNA, without this chromosomal DNA. Okay, no issues, beta. See, this along with the main DNA, you have plasmid. Okay, yes. So what is the importance of plasmids in bacteria, children? These plasmids carry some important genes. These plasmid carry some important genes like, antibiotic resistance gene. So this makes the bacteria resistant to antibiotics. That means this plasmid containing some genes which make some protein. The protein protects the bacteria against antibiotics. So whenever an antibiotics comes near the bacteria to kill it, the proteins made from bacteria just give a protection, a protective cover, and it shields the bacteria and protect the bacteria from antibiotics. Okay, so this is all I wanted to mention about plasmids. Now going back to your previous slide where we were discussing about vectors as plasmid. Yes, here. So now you have fair idea like what is plasmid. Okay, so for children who didn't know about plasmid, I just gave a brief introduction. Now see, I have plasmid. Okay, what I do, I use restriction enzyme. I digest the restriction enzyme. Can you see here? Yeah, and with the same restriction enzyme, I cut the for my gene of interest, that is insulin. Now with the help of ligase enzyme, I inserted, I fixed, or I glued that insulin gene into the plasmid here. And now what I do, I give it back to E. coli cells. Okay, so this is E. coli cell. So now I transfer this plasmid into E. coli cell. Okay, so now this is rDNA, right? Whatever the E. coli is having is its rDNA, and in addition, it is having its main DNA. That is different. Okay, so now your E. coli is recombinant cell, right? Yes. So now coming to use of phages, viruses as vectors. You can see this is your target gene. Now what you do, you pack the viral particle with your target gene, and now you infect the cells with your target gene carrying virus. Now virus will release that genetic material into host cell. Now the host cell will take up the genetic material, and inside the host cell your foreign DNA start expressing, right? So this is how we can use either plasmids or phages as vectors to carry a foreign DNA or gene of interest from one cell to another cell, children. Okay, any doubts here? Children, any doubts till now? Any doubts? Any doubts, children? I'm looking at QA panel. If you have any doubts, just let me know. Okay, I assume you don't have any doubts, and so I'm moving towards next slide. Okay, so now you know what are vectors, what is the importance of vectors, and you also know how to make recombinant DNA, right? So children, see, now I'm just giving you conclusion part, like how we use restriction enzyme and how we made the recombinant DNA, right? So you know that foreign gene is there, plasmid is there. I'm using restriction enzyme to make the fragments. So using ligase enzyme, I made the recombinant DNA, right? Yes. Now I have to transfer this recombinant DNA into E. coli, right? This process is called transformation, children. This is very important word, transformation. What is transformation? Transformation is a process. Transformation is a process where a foreign DNA or extracellular DNA will be taken up by bacterial cell. Foreign DNA will be taken up by bacterial cell. Can you see here? Transformation is a process where a foreign DNA, see, now this is your rDNA, right? It is a foreign material for E. coli. It is not its own. So now I am making this rDNA to enter the E. coli cell. So your E. coli cell is uptaking the extracellular DNA. Okay, this process is called transformation. This process is called transformation, and the cell which takes this foreign DNA is called recipient cell. Recipient cell, also called competent cell. Competent cell means the cell which is ready to take the extra DNA, like my students in this class are competent students. What is meant by competent? Yes, they are having the ability to take whatever I'm teaching them. Okay, competent. You should be competent. Competent means what? So you should take up the things inside you, right? Yes, so the same way here, the bacteria which is taking the foreign material is called competent bacteria. Okay, yes. So let us study more about transformation, how you are making bacteria cell to take up DNA. Okay, so sometimes you use chemicals, sometimes you use some instruments, and sometimes it is natural. Okay, yes. So in your textbook we are coming with chemical method and some physical method. So I'm going to elaborate more on this concept. Okay, using chemicals, how to make E. coli cell to take up the rDNA into it. Okay, yes. See, first one is chemical method. Here what I do, in a test tube, imagine as or assume, okay, now your body's test tube, a reaction is happening inside test tube. Now you have your E. coli cells. Okay, this is E. coli cell. Okay, and these are your plasmid with your foreign gene, nothing but your rDNA. Okay, yes. So now you need to make this rDNA to enter your E. coli cell. How you're going to do? Cell will not take up easily. Okay, so what you have to do, see, you will study when exams are ahead, so the pressure should build up and then you will study, right? The same way here, you should give some shock. Okay, you should give some shock such that the permeability of the bacterial membrane will increase, and in that way the plasmids can easily enter into the cell. So what kind of shock will do, we'll see here. We mix E. coli plus rDNA plus calcium and we keep it on ice. So we take a test tube here. Imagine it is a test tube. In this test tube, what all you're adding? You're adding your E. coli, okay, and you're adding your plasmid with your foreign DNA, right? And you're also adding calcium. Okay. Now what you're doing? You're putting the test tube in a mug with ice cubes. So you're keeping the test tube on ice. So now the bacterial cells inside the tube feel shock, cold shock. Arey babre, kya hua? Like temperature is suddenly going down. So it gets shocked. Now we need to give more shock. Okay, now you're feeling cold. Okay, now see what I'm going to do to you. I'm suddenly keeping the test tube in hot water, a beaker with hot water. Now what will feel bacteria? Arey baba, kya ho raha hai? Just 1 minute back I'm feeling too cold. Now I'm feeling too hot. So what I have to do? It get confused. So it start, protecting itself. During that time, the membrane permeability will increase. Membrane permeability will increase. So this sudden cold and heat shock in the presence of calcium will increase the membrane permeability. Okay, so this process increase membrane permeability of bacteria. Membrane permeability. Okay, so as the membrane permeability increases along with the calcium, so your plasmids or the rDNA will take up by the bacteria. So this is how, using chemicals like calcium, we are going to make the bacterial cell more competent to take up the foreign DNA. Okay, yes, I hope this is clear for you. I hope this is clear for you, right? Yes. So now I'm going to show you, discuss more about physical method. Physical method to make the bacterial cell take a DNA. Okay. So first method is micro. There are two methods. There are actually many methods, but according to your syllabus, we are discussing two methods. Okay. Two methods. So one is biolistics or gene gun method first, okay, and second one is micro injection method. Okay, so first we'll take a biolistic method. What happens in biolistic or gene gun method? See the, look at the name, gene gun. Okay, what we are going to do, these are gold particles. Okay, now you need to dust the rDNA onto gold particles. Can you see here? So you need to load the DNA, you need to precipitate the DNA onto gold particles, children. Yes. And now you load those DNA, rDNA coated gold particles into some long tubes. See? Now you're rotating the tube just to make them move. Okay. And now these are using as bullets in gun, children. Okay. These are called cartridges and these are like bullets. Now see, just load the gun, gene gun. This is called gene gun. Okay. Now load this gene gun with these gold particle which are coated with rDNA cartridges. Okay. And then shoot at particular cells. By doing this, you're going to deliver rDNA into the target cell. So this is one of the physical method. Okay. Yes. So another one method is micro injection. So here what we're doing, we take up the rDNA particles in a fine pipette wala syringe, and then we take cell, we hold the cell firmly using a pipette, and then we directly inject those into the cell with the help of needle, fine, very fine needle, we directly inject into the cell. So this is one more physical method to deliver the rDNA into a host cell. Okay. Yes. So these are the different transformation methods, children. Either you can go for chemical or you can go for physical. In chemical, you use calcium along with heat shock and cold shock treatment. In physical method, you use biolistics, that is gene gun, or you can also go for micro injection. Okay. Yes. So this is all about transformation of rDNA, recombinant DNA into cell. Okay. So with this, we completed second tool, right? That is vector, and how to use it and how to transfer that vector carrying foreign DNA into bacteria. And also we discussed about ligase, right? Yes, children. Now we are going to start, a little more exploration about vector. Okay, cloning of vector, cloning of vector. What is cloning of vector? See, a cloning vector is a vector vehicle that carries your foreign DNA into it and delivers it to a host cell, right? So it is for different purposes. It is for different purposes, right? Yes. So now let us see what are the features, what are the characteristics should be there if you has to claim some DNA molecule as vector. What are the characters you need to find in them? Okay, what are the requirements for the vector to clone? Okay, yes. So in this slide, we are going to explore more on the features of vectors which are used for cloning. Okay, so here I'm taking an example of vector plasmid. Okay, here I am taking an example of vector, that is plasmid. There are many plasmids, right? We have isolated so many plasmids, but in this I am picking up the plasmid. The name of the plasmid is PBR322. Yes, so we are taking this as an example and we are learning the features of cloning vectors. So what are the essential features one vector should have? Okay, yes. So first let us study what is this PBR322, ma'am. It's more like, abbreviation. What it is? Yes, so PBR322 is a type of plasmid, which we commonly used as vector in E. coli. Okay, so here PBR means what? P refers to plasmid, BR are the scientist name who discovered this plasmid. Okay, Bolivar and Rodriguez. Bolivar Rodriguez, BR. Plasmid discovered by two scientists, Bolivar and Rodriguez. Yes. And what is the number 322? This plasmid model was the 322nd invention in the lab where they were working. Okay. So look at how many models they have developed. Okay. They are wonder, right? Yes. So these two scientists, Bolivar and Rodriguez, they discovered this plasmid from E. coli, and it was the 322nd model developed in their laboratory. Hence, as a recognition, they gave the number 322. So this is how this plasmid got the name PBR 322. Okay, so any doubts in the name of the plasmid? Any doubts in the name of the plasmid, children? Are you getting any doubts? No. Okay, it's clear. Now look at the plasmid picture here. It is having some important, labeling and important features in it. Okay. Yes, children. Imagine you're starting your two-wheeler. Okay, you're starting your two-wheeler, any vehicle. You want to do some start. So what you do? You just kick that engine, right? Why you're doing that? You need to ignite the engine. You need to initiate, right? The same way, to initiate any process, you need some point of initiation, right? You need to some point of initiation. That means, thank you, Avinash. Thank you, children. Thank you for your wonderful responses. Yes. Yeah. Say, for example, you're writing on notebook. From where you write? Can you write from the last page or from middle of the page you start writing? No, you will start from first page and first line, right? It is your initiation point in your notebook. The same way here for plasmid, plasmids are replicating, right? Plasmids can do replication. So if they are doing replication, from which place they have to do replication? Look at this plasmid structure. Can you see here this fragment? Yes, it is called Ori site. Ori site. What it is, Ori site? Or Ori site is nothing but it is the point of origin of replication. So it is the place where replication is taking. It is the place where replication is taking in the plasmid. Okay. So whenever this plasmid do replication, that replication will start from this point. So that's why Ori site is very important place in the plasmid. Okay. Yes. So if you observe carefully, next to ORI, you have some place called ROP. Okay. Yes. So this is nothing but repressor for primers. Repressor for primers. Okay. So don't worry much about this word, children. Just know this. The full form of ROP is repressor for primers. What is the function of this site? It is going to regulate the copy number. Copy number in the sense, see, a bacteria can have plasmid and this plasmid is undergoing replication. That means inside the bacterial cell, the plasmid is, replicating. It's increasing in its number. So how many plasmids are there? See, this is the original plasmid. From this original plasmid, so many replications happen. So here, this is copy number. So this will be regulated by ROP, repressor for primers. Okay. Yes. And then apart from this, this plasmid should have cloning site. What is cloning site? What is cloning site, children? It should have some place to insert foreign DNA, right? Here and there you cannot insert. You should see there is a bus. You step into a bus. Can you go and sit everywhere, anywhere in the bus? No, you have some seat. You have some place to sit. So you go and sit there. Similarly, inside your class, you don't sit randomly in the class, right? You have some specific place. You go and sit there only. Same way, in this plasmids, you have some specific places where your restriction enzyme can break the plasmid, and at the point of breaking you can fix your foreign DNA. Yes, you can see here this PBR322 plasmid is having so many cloning sites. Can you see cloning sites? Nothing but the sites where restriction enzyme can cleave the plasmid and insert the foreign DNA. Okay, I repeat, cloning sites are the sites where restriction enzyme C EcoR1, PVU1, PST1, BAMH1, SAL1. So all these are, restriction enzymes. So they are having some specific recognition sites. Say for example, for eco R1, it is having here GATC, right? GAATTC. GAATTC. So it is having recognition site here. So now eco R1 cuts here, and in this place you're going to insert your foreign DNA, right? You're going to insert your foreign DNA, right? Yes, so that's why these are cloning site, and a plasmid can have more than one type of cloning site, and it's your wish, it's your choice which type of restriction enzyme you're using and where you're inserting foreign DNA. Say for example, if you're using eco R1, then you're inserting your gene, your foreign gene. You're inserting your interested gene at eco R1 site. If you are using PST1 enzyme, then you're cleaving the plasmid here and inserting your foreign DNA here. If you are using, SAL1 restriction enzyme, then you're cleaving here because enzyme is having recognition site at this point. You're cleaving the plasmid and you're inserting foreign DNA here. Okay, yes, so that is the beauty of PBR322 children, because it is having more restriction enzymes sites such that you can use you have many options, right? So for example, which restaurant you would like to go always, where you get different menu, right? If a restaurant gives only idli all the time, maybe sometimes you go, but not all the time. You're not in a position to eat the same food, so you need varieties. Sometimes you have more money, so you want to eat more delicious food. Sometimes you have less money, you want to eat less food with a less price, right? The same way, depending upon your DNA. See, your plasmid is having eco R1 restriction site, but it doesn't mean the gene which you're isolating, your gene of interest, also should have recognition site for eco R1. No, it's not compulsory. It may have recognition site for SAL1. So then which restriction enzyme you need to use? You need to use SAL1, because you're having SAL1 recognition site in your desired gene as well as your plasmid. So this is how you need to choose the plasmid restriction enzyme and the gene of interest in combination. Clear, children? Clear? Up to here, any doubts, children? Up to here, any doubts? I'm just looking at your QA panel. Up to here, any doubts in the features? No. Thank you so much. Okay. So now observe carefully. We are studying silent features of this plasmid, right? Yes. So we studied what is origin of replication. We studied what is ROP. We studied what a cloning site. Okay. Yes. So now if you observe carefully, you also have some labels. Can you see? AMP, ampicillin resistance. TET, tetracycline resistance. What are these? What are these? These are also characteristics of plasmid. These are also characteristics of plasmid. What are these? Let us study. Let me take you to next slide where I can explain this concept in better and clear way. Yes, so we already studied the features like origin of replication, ROP, and the restriction enzyme recognition or cloning sites. Okay, now we're left with these. What are these? These are nothing but marker genes. marker genes are also called selectable markers. What is the function? What is the function? Say, for example, AMPR. What it means? What it means? It means ampicillin resistance. That means this gene makes some protein. Ampicillin gene is somewhere here. Yes, it's overlapping the genes with, sorry, nucleotide sequence with cloning sites. Okay, it can happen. See, ampicillin resistance gene somewhere here. This ampicillin resistance gene will make some protein. Okay, this protein is going to protect the bacteria against ampicillin. Same way, tetracycline resistance gene. Somewhere it is here having gene. This gene is, making some protein. That protein makes the bacteria protective against tetracycline. What is the advantage of this? Why the plasmid should have these resistance gene? How can we exploit that? Okay, see. Let us see these two cells. This is non-transformed cells. Why it is called non-transformed cells? Because no rDNA, right? No rDNA. No rDNA means there is no plasmid and there is no foreign DNA, right? There is no plasmid carrying foreign DNA is here. Hence, the cell is non-transformed. Here, the cell has taken plasmid. Yes. Along with the foreign DNA. Yes. So now this is transformed cell. Children, this is what I am showing you on slide. But in real lab, you have both the cells in mixture. See, this is your transformed cell and this is your non-transformed cell. This is your transformed cell and this is your non-transformed cell. So you're having a mixture of these two. Now, which one you want? Which one you want to take up for your experiment? Which one you want to take up, children? Tell me. Would you like to take up only transform cell or both transform and non-transform cells? Which one you would like to take up? Come on. Come on. Come on, children. Respond to me. Yes. You want only transform cells, right? You don't want non-transform cells because it's waste. Why you're investing on that? So you want to take up only transform cells. Now, how you're going to choose it smartly? I'm going to tell you. I will make it media in a petri plate. This is your petri plate. Petri plate means a glass dish which is used to grow microbes. Okay. So now I'm taking a petri plate, a glass dish, and I'm pouring a mixture of both transform cell and non-transform cell. I don't know which is transformed, which is non-transformed. So I'm just pouring that solution containing both the cells into this. And to this petri plate, I have added ampicillin, antibiotic. Ampicillin is an antibiotic, right? So I have added ampicillin antibiotic. Now, as these non-transformed cells are not having any plasmids, they're not surviving. Why? See, the transformed cells are surviving. Why? They have plasmid. Which plasmid they are having inside it? So they are having PBr322 plasmid. So what this PBr322 is having? PBr322 is having ampicillin resistance gene in it. So as it is having ampicillin resistance gene, when this bacteria is growing in ampicillin medium. Somewhere here, the ampicillin resistance gene in this plasmid is making protective proteins. So this protective protein giving resistance to this cell to fight the ampicillin. That's why these cells are surviving. Are you getting, children? Are you getting? Yes, W beta, I'll come back to your question after this slide. See, are you getting? Why the transform cells are surviving? Because transform cells are having plasmid like this, a PBR 322 plasmid. This plasmid is having some antibiotic resistance gene. The antibiotic resistance gene will give some protein, some chemicals that will protect the cell from antibiotic ampicillin. That's why in the petal plate, only transform cell which are antibiotic resistant will grow and non-transform cell will die. So whatever you take from this are purely transformed cell, children. Whatever you're getting on this plate are purely transformed cells. So this is how you're going to select transform cells. You're going to select only transform cells from the mixture of both non-transformed transform cells using this antibiotic resistance gene. Okay, now this antibiotic resistance gene is helping me to select transform cells, right? Hence, these are called selectable markers or marker genes. Is it clear, children? Do you get how I'm going to exploit these antibiotic resistance genes as selectable genes to select only the recombinant cells? Why they are called selectable markers? Because they are helping me to select only the recombinant cells, only the transformed cells. Okay, hence the name to these genes in plasmid, we call them selectable markers. Any doubts, children? Any doubts here? No doubts. Yes. Yes, W beta, while I was explaining some basics about PBR plasmid, you are just going on and off due to some issues maybe. One minute, beta. I'm just changing the slide. Sorry, it happened randomly. Yeah. So PBR history you wanted to know? Yes. W beta, this slide is for you. As you requested, I am giving you full information on nomenclature of PBR 322. P for plasmid, BR refers to scientist who discovered it, Bolivar Rodriguez. Okay, 322. Beta, Bolivar and Rodriguez were working in a laboratory. In that laboratory, the discovery of this plasmid was the 322nd discovery. Hence, they label it as 322. So, plasmid, Bolivar, Rodriguez, 322. 322nd model developed in that laboratory. Okay. Yes. Okay. So can I go to the topic what I was discussing? Yes. So we were discussing marker genes. We were discussing silent features, right? So now you know the important silent features of a plasmid or a vector. So it should have origin of replication, Okay, such that as it start replicating its plasmid DNA, along with that, it will take your inserted DNA in the plasmid also to replicate. And you should have copy number gene, and you should have cloning sites. So plasmid or an vector ideally should have more and more cloning sites such that you get better options. Okay, yes, and you should choose the cloning site depending on the recognition sites in your gene of interest also. Okay, in your foreign DNA, say for example, if I'm taking insulin, in insulin, among these restriction enzyme, which restriction recognition site is available, no? So considering that, I need to choose the cloning site here. Okay, if my foreign DNA is having recognition site for EcoR1, then I'll choose this site here. If my foreign DNA is having recognition site for BAM-H1 restriction enzyme, then I'll use BAM-H1 to cleave foreign DNA as well as plasmid, and I will insert the gene here. Okay, so this is how we need to choose the DNA and restriction enzymes carefully. Okay, yes. So next, I hope you understood the silent features of Plasmid children, and also selectable marker is also another important feature of plasmid, and we use this as smart trick to choose only the transform cells after our experiment. Okay, yes. So any doubts till now? Any doubts, children? If you don't have any doubts, okay, shall we do some questions? So depending like what all we discussed so far. Okay, thank you, beta. Okay, so shall we do some quiz? Okay. Yes, tell me. In last session we discussed about this, no? EFB. What is EFB stands for? EFB stands for option A, European Federation of Biomolecules, European Federation of Biotechnology, Europe Fertilizer Board, or environmental friendly biomolecules. Very good, children. Very good. I'm very happy. Okay, thumbs up from my end. For all of you, thumbs up. Yes, it is B option, right? Yes, European Federation of Biotechnology. Always remember, you have definition of biotechnology proposed by European Federation of Biotechnology, right? So you know the application or integration of natural science along with the molecular biology, cells, genetics, and all to get the products from cell parts, tissues for the betterment of mankind. So this was the definition given by European Federation of Biotechnology, right? Yes. So next, let us see what is there for you. Okay. Now see, next question. Question is, the specific base sequence recognized by restriction enzymes called gene sequence, recognition sequence, numerical sequence, or all the above? Wow. I think you guys enjoy more and more quizzes. Okay, we'll do one thing from next session. After every concept, let us do some quiz. At some 20 minutes break, after some 20 minutes of concept, let us do some 2 to 3 quiz questions. I see more students like all of you loving to answer quizzes. Yes. Okay. It is recognition sequence, right? Good. So answer is Recognition sequence. Gene sequences can be any nucleotide sequence, right? So recognition sequence is a sequence of nucleotides recognized by restriction enzymes. Good. Okay. So now next question. Let us see what is there in next question. Yeah. Which of the following are palindrome? Which of the following are palindrome sequence? GATAG, Malayalam, ATATA, all of the above. Tell me. Tell me. GATAG, Malayalam, ATATA, all of the above. What is palindrome sequence, children? See, I'm telling you, any word, to and fro. To and fro should be same, right? Yeah. See, this is a general question, okay? So always whenever you do some competitive question, you should have some common sense. Yes, I know. I'm not asking you which of the following gene sequence are palindrome sequence. The question was which of the following is palindrome. GATG, can you see here? This one. To and fro, back and fro. Same word. Malayalam, same. Or Tata, see, same. So all the above are palindrome. Yes, children. Yes, good job. Good job. Okay, so this question I put to confuse you. Okay, you will be thinking only about gene sequences. Look at the question. It's palindrome. Pattern I'm asking. Okay, not an issue, beta. Not an issue, W. Okay. So all the best. Now let us look into, yes. Now here you have a question. The recognition sequence of echo R1, GAATTC, GGCC, AAGCTT, or CTGCAG. Which one is the recognition site? Wow, I'm getting wonderful response. Good, good. Good going. 100% result. Yes, 100% of you are giving right answer. Yes, always remember this is very important question even from your NEET point of view. GAATTC is the recognition site for echo R1. Yes. So any doubts, children? Do you have any doubts from this question? If not, okay, let us see what is that for you in the next. Yes, it is the end of the session, right? Okay. Yes, we came to end of the session. We are ending session here. I hope all of you enjoyed the session. In case if you have any questions, children, please do drop your questions in the question box. Even after that, I'll take your questions and I try to give answer in next session. Okay. And if you are, if you want to tell me something, please comment in the box and please do like the sessions, share the session, help your friends to know about my teaching, share this session link and help others as well. Okay, yes children, please do like and share and come back tomorrow for next session. And in next session we are going to discuss the last part of this chapter, that is how to make products. So now we know all ideas, we know tools, we know principles, we know how to use vectors, how to choose the vectors, and how to choose restriction enzyme for which type of DNA, which type of restriction enzyme need to choose, and which type of vectors we need to select. All so now you have ingredients. Now you need to mix the ingredient in an order to make a beautiful delicious dish. The same way tomorrow we are applying all the knowledge and concept what we discussed in 2 sessions, and we are coming to a conclusion of our DNA technology, and we are concluding the chapter with more and more quiz questions. Okay, yes children, till then take care, bye bye.